Greetings and welcome to another lecture. Today we're going to talk about how electrons are transporting in electron transport chain in mitochondria. So let's get started. This is our mitochondria and it has two membranes, outer membrane and inner membrane. The space between inner membrane and outer membrane is called IMS or intermembrane space, and the space within the mitochondria is matrix. So let's review what happened from the beginning until here. We talked about it before that we imported a glucose into the cell, and glucose can do glycolysis in order to turn into pyruvate. Pyruvate in aerobic condition can enter mitochondria here, and it eventually can turn into a 2-carbon molecule that we call acetyl coenzyme L. Acetyl coenzyme A eventually can follow Krebs cycle pathway and produce plenty of different molecules such as NADH and FADH₂. And now in this lecture we want to see how the mitochondria use NADH and FADH₂ in order to produce energy. These guys, NADH and FADH₂s, are hydride carrier. Totally, we can say they are electron carrier, but more scientifically, we can say they are hydride carrier. Hydride is a hydrogen with additional electron. Normally, hydrogen has one electron, but hydride is a hydrogen with one additional electron. So we can write like this: hydrogen plus 2 electrons. It's a hydrogen with 2 electrons. Instead of 1, it has 2 electrons. So in NADH case, this little body can comes here and donates its electrons into this structure. This structure has a molecule called FMN, flavin mononucleotide. This molecule receives electrons and turns into FMNH2, and this molecule is not stable now. It has a lot of energy, it has electron on its own. So he's trying to find a way to get rid out of these electrons. What this little guy do? He finds a neighbor here called Q. Q can sacrifice himself and receive his electrons and turns into QH2. This is what happened for NADH. In FADH₂ case, there is another structure. The structures are not the same. It has another structure, and it donates its electron to this structure and turns into FAD. There is a molecule just like here that we call FMN, and this beautiful molecule receives electrons in order to turn into FMNH2. FMNH2 is not stable. He wants to get rid out of these electrons, so he is trying to find neighbors. He finds one and he donates his electrons into Q, and Q turns into QH2. So what is going on here and what happens? This structure we called NADH dehydrogenase, or totally complex number 1. These structures, these neighbors, are coenzyme Q10, and this green structure that is responsible to receive electrons from FADH₂ is succinate dehydrogenase. Succinate dehydrogenase is useful in Krebs cycle 2, if you remember. And totally we call this structure complex number 2. Let's see what happened here. This is a diagram. We have delta E and reaction progress. In the case of complex number 1, this is our delta E before receiving electrons from NADH. After receiving electrons, it goes up, and now it has a lot of energy because if MnH2 is not stable, it has a lot of electrons and energy, and after donating his electrons to Q, the energy amount drop. What happened here and what is important here, that complex number 1 has some specific pumps. These pumps can pump out hydrogen into intermembrane space. Let me tell you again. There are hydrogen pumps that are located on NADH dehydrogenase on complex number 1, that they can export hydrogen from matrix into IMS. For complex number 2, it's somehow the same. The energy level is here. Whenever it receives electrons, the energy level goes up, and whenever it donates its electrons, the energy level drops. But you should know that in contrast of complex number 1, complex number 2 does not have any hydrogen pumps, and this is very important to know. Also, you should know that coenzyme Q10s are movable. They can move on inner membrane, and they are not stable. They can move whenever they want on inner membrane, and they can find FMNH2s in order to receive their electrons. So now we have many QH2s actually on inner membrane space. So we have another structure here. We call this structure complex number 3, or scientifically we call this Q, or coenzyme Q, cytochrome B oxidoreductase. This structure has cytochrome B, and it is responsible to receive electrons from coenzyme Q10s. It is responsible to receive electrons from QH2, and it has Fe3 positive. And whenever Fe3 positive receives electrons to sacrifice himself, he turns into Fe2 positive. On the diagram, this little dude also is in a stable state. Whenever it receives electrons, the delta E goes up, and it drops whenever it donates its electron. And this guy, just like this guy, has hydrogen pumps. You should ask yourself, why do we need these pumps? Totally, the amount of hydrogen in matrix is this number in millimole. And in normal situation, the number of hydrogen in intermembrane space is the same as matrix. But whenever we kick out these hydrogens, it is logical that the hydrogen level, the hydrogen density inside IMS increase. That we will talk about it later. So we have now Fe2 positive. Fe2 positive is carrying electrons, and it is high level energy. It has a lot of energy, and it wants to donate his electrons. So we have another structure here. This is cytochrome C. Cytochrome C, just like as complex number 3, has Fe3 positive. It can receive electrons and turn into Fe2 positive. It is important to know that this structure is not complex. This is cytochrome C. We don't call this one, for example, complex number 4. This is cytochrome C, and this one, this marronish structure here, is complex number 4. So Fe2 positive now has a lot of energy, it carries electron, and we have cytochrome oxidase here. Cytochrome oxidase, or complex number 4, has cytochrome A plus A3. This guy also has Fe3 positive. It can receive electron in order to Fe2 positive. But there is a difference. Whenever electrons are in this step, this complex number 4 can use exactly oxygen atoms, can use oxygen atoms to produce what? H2O or water. In other words, the final electron receiver in electron transport chain is oxygen. And oxygen is available whenever we have electrons on complex number 4. Fe2 positive donates his electron to oxygen, and it uses also hydrogen in order to produce water or H2. In the diagram, it's just the same as the other. And it is important to note that this structure, this complex, also has hydrogen pump. Now we export a plenty amount of hydrogens out of the matrix, and now the amount and the density of hydrogens in IMS is this number, and this number is higher than this number. This number in millimole is higher than this one. So there is a difference in their concentration in inner membrane space and in matrix. In the next video, we specifically talk how this difference can start this engine in order to produce ATP for us. I hope you really enjoyed this video. Please ask any question if you want on the comment section. There are some additional information on description section, and also you can have these illustrations if you want for free on Telegram channel, which the link is below in description. So good luck and see you until next video.